1931, German physicist Ernst Ruska and electrical engineer Max Knoll built the first transmission electron microscope. At the time, it's not as good as optical microscopes. In the later 1930s, at the University of Toronto, James Hillier and Albert Prebus build on the work of Ernst and Knoll to create a transmission electron microscope capable of a magnification of 7,000-fold. That's approximately 3 to 4 times better than any other microscope available at the time. James Hillier joins the Radio Corporation of America, RCA, in 1940, and he works with Vladimir Zworykin. They develop a small, affordable electron microscope capable of 100,000-fold magnification. And in 1941, they demo it, and that's the picture you see. A couple years later, McGill University acquires that same model of electron microscope. And in the late 1950s, that microscope gets transferred from the Eaton Electronics building into the Strathcona Anatomy building. The Facility for Electron Microscopy Research, or FEMR for short, is an open-access electron microscopy facility at McGill University. FEMR is located in the basement of the Strathcona Anatomy and Dentistry building, right beside the University Street entrance. In this short video, we're going to walk you through all of the equipment relevant to a NAT 542.
The equipment that you see here is a carbon coater. We typically use this to produce a continuous carbon layer on the grids. For the purpose of cryo-electron microscopy, the continuous carbon layer is essential to prevent proteins to get exposed to the air-water interface, which causes protein denaturation. And for the purpose of negative staining, this equipment can produce a carbon support film on which your molecules of interest will adsorb. This is a Technique G2 Spirit transmission electron microscope and is capable of providing high resolution images. The beauty of this microscope is that it is very easy to use and it's also very instrumental in visualizing biological samples that are sensitive to radiation damages from a higher dose of electrons. Using this microscope, several research groups can visualize the sections of tissues and cells, allowing them to visualize the histology of the cells in an unprecedented detail. Using a technique like negative staining, it is possible to visualize macromolecules and even tiny components of cells such as individual protein with great contrast. Usually, negative staining is the first step towards successful structure determination of any biological components using cryo-electron microscopy. The microtubules that you see on the screen are a great example of negative staining. The equipment that you see here is a glow discharge and is an essential component of the sample preparation. The glow that you see in this video is generated by the ionization of a low pressure gas, which in this case is air itself. This allows us to remove the hydrophobic contaminants on the surface of an EM grid and makes the spreading of the sample on the grid in an even manner. The next equipment that you see here is a Vitrobot and is used in plunge freezing of the biological samples. The plunge freezing allows us to trap the protein or other macromolecules in a vitreous ice that enables us to visualize them in a near native state. In 2017, Jacques Dubochet was awarded Nobel Prize for inventing and developing the technique of plunge tracing. Once you prepare the cryo-EM grids using this equipment, you are ready to visualize them under a TEM microscope. This is a Technia F20 microscope and is much powerful than the Spirit microscope that you saw earlier. We typically use this microscope to screen biological samples of interest in vitro size. This microscope is currently equipped with a CMOS technology camera and is able to read electrons to produce high-resolution images. For viewing the biological samples under cryo conditions, we typically use a cryo holder on which we place our grid and then transfer the cryo holder to the microscope for visualization. Using this microscope, we can determine the quality of the sample preparation by evaluating ice thickness, protein distribution, and overall integrity of the grid. If the grid passes all good criteria for the sample, we then generally proceed for high throughput data collection for structure determination using the Titan Cryos microscope. This huge microscope that you see here is Titan Cryos and is the most powerful and flexible high-resolution electron microscope for 3D characterization of biological samples. This microscope is typically used to collect movies for protein structure determination, reconstruction of cells, viruses, macromolecules, and other cytoskeletal elements such as microtubules. During the COVID-19 pandemic, this microscope has been the key to visualize the structure and integrity of several vaccine samples, allowing further development of effective vaccines. This microscope allows us to collect high throughput of data, and to give you an example, it can collect approximately 8,000 movies in a day. To put this number into a perspective, a well-trained human can take approximately 500 to 1,000 images in the same period of time. That is how complex the microscope looks from inside. At the bottom here in blue, you have this amazing camera which is a direct electron detector, and as the name suggests, it can record the electrons scattered from the sample directly, allowing us to take very high resolution movies which will eventually help us in reconstruction of structures with details at atomic level. Since the microscope is sensitive to vibrations, it is usually situated in a dedicated room and is controlled from a different room. The Helios NanoLab 660 is one of the most advanced field emission scanning electron microscopes that is combined with the focused ion beam technology. Using this technology, we can prepare the thinnest and the highest quality samples of cell sections that can be used for tomographic reconstructions.
Ultramicrotomy is a sample preparation method for predominantly transmission electron microscopy. Biological samples are embedded in plastic resin or frozen at cryogenic temperatures, at which point they are roughly cut into blocks. Those blocks are mounted onto the ultramicrotome, and using a glass blade, the blocks are cut until the desired section is less than 2 micrometers thick. And then the glass blade is replaced with a diamond blade. And the diamond blade is then used to cut ultra thin sections of about 50 nanometers in thickness, at which point they're mounted onto copper or gold grids like traditional electron microscopy, and then observed under a transmission electron microscope.